So we have now finished up translation. Everything has fallen apart and the ribosome has released the polypeptide chain. So that's what we have here. These purple little shapes are our amino acid chain, and they are linked together to make what we call a polypeptide. If you remember from chapter 3, I know, the dreaded chapter 3, when we link together amino acids, we produce a peptide bond. Now, an amino acid chain has a direction, just like a messenger RNA. One end is called the N terminus, that's the end with the free nitrogen. The other is called the C terminus, the end with the free carbon molecule. And when you add an amino acid to the the carbon end of a molecule and you link it with the nitrogen end of the molecule, you produce a peptide bond. So our amino acids are the monomers linked by a peptide bond. And remember, this is done by a dehydration reaction where we produce a water molecule that is released from the production of that bond. So when we finish up translation, we often say we make a protein, but that's not entirely true. We actually make an amino acid chain, and that is the end of translation. We have this long chain of amino acids. Before it can actually be functional, we have to fold it into a protein. We have to get that protein into its proper 3D shape. So remember, going back to the beginning of the semester, structure equals function. So the protein needs a very specific shape in order to perform its very specific function. So if you remember from chapter 3, there were several levels of protein structure. And I'm not going to go into too much detail on these. This is really just meant to be a quick review to discuss protein folding. The primary structure is what comes out of the end of translation. That is your amino acid chain. So we can see this long amino acid chain. This is for a protein called transthyretin. Which is really not important for our discussion. It's just an illustration of the amino acid chain. So we have this series of amino acids. Each one was coded by the codon for that particular amino acid. So this first one, this glycine, is showing the set reading frame. So glycine, proline, threonine, etc., all the way down. If we had a change in our reading frame, everything would shift and be a little bit different, and we'll see why in just a minute. After the primary structure, we need to make a 3D protein. So once the ribosome begins to release the growing polypeptide chain, it actually begins to fold spontaneously. And that happens due to interactions between the different groups off of the amino acids, the R groups. Some interactions occur between the backbone or the frame, the nitrogen carbon backbone, and some interactions occur between the R groups that are coming off of them. Once all of this folding is complete, the amino acids are usually modified chemically, so they're sort of like activated and tagged and things like that, so that the cell knows what to do with them. All of this happens in the, all of this folding happens in the endoplasmic reticulum or in the cytosol of the cell, depending on where that ribosome actually is. This ribosome is shown attached to the endoplasmic reticulum. And then the protein is further modified in the Golgi apparatus, and that Golgi apparatus is then going to package up that protein and take it in a vesicle to the edge of the cell, and it's going to be released. So this amino acid chain is going to, this primary structure is going to fold back onto itself to make a 3D shape. So some of those shapes are due to hydrogen bonding between the R groups on the amino acids, and we get two different types of shapes. We get the alpha helix or the beta pleated sheets, and the presence of the amino acids, the sequence of the amino acids, is actually what's going to determine whether or not the alpha helix or the beta pleated sheets are formed. From there we get tertiary structure, which is the overall folding of the protein. So we have these helix and sheet regions are sort of folded back on one another and bound together by covalent bonds that are going to hold that protein structure tight. Some proteins have multiple tertiary proteins that are stuck together to make sort of one big protein. So let's talk about a specific example of a change in the nucleotide sequence that leads to changes in proteins. So an example of this is sickle cell anemia. The function of our red blood cells is to carry oxygen throughout the body. So the red blood cells are filtered past the lungs where they pick up fresh oxygen that you breathe in. They carry it to muscles or organs, wherever that oxygen is needed, and then the oxygen is dumped off there. But in the disease sickle cell anemia, you actually have misshapen red blood cells. And this occurs because of a single base pair change in the DNA sequence that causes a change in the amino acids. So when we get to our lab this week, we'll actually look at what the actual DNA change is, and we'll start at the DNA sequence. But for now, our book shows us that amino acid 6 in this sequence is supposed to be a GLU amino acid. That is a glutamine. And instead, the DNA sequence directs it, if you have this particular allele, to be a valine. When that happens, instead of getting this nice round normal subunit from the quaternary structure, you get this little divot in the side of the cell. So normal hemoglobin has 4 subunits. They're all nice and round, and when they are nice and round, they interact in the correct way, and they form the hemoglobin inside of the red blood cells. But instead, in sickle cell, when we have this little divot here that is caused from the change in the protein sequence, we get these misshapen subunits. What happens with those subunits is, instead of staying nice and round and not interacting with one another, they actually bind to one another and produce these long fibers of red blood cells. Now, this is a problem because when these fibers are formed, you're not carrying oxygen throughout your body. So oxygen cannot correctly bind to the hemoglobin inside your blood cells, and therefore it's not being carried correctly. A side effect, sort of those fibers forming is that it causes the blood cell, the red blood cell, to be sickle shaped. Now, this is not normal. It should be this nice round plump shape here. But those fibers, that interaction with those hemoglobin molecules, actually produce this sickle cell. Now, this causes a lot of effects in the individual besides just the lack of oxygen capacity. It also, these sickle cell blood cells can accumulate in organs. They can get stuck in arteries and cause a lot of pain for the individual. And at some point, a lot of times the organs begin to shut down. So this is an example of pleiotropy, which was something we discussed in chapter 11, which is where you have one specific gene that has multiple phenotypic effects. So not only do we have the reduced oxygen capacity and the physical change in the hemoglobin, but we also have that change in the red blood cell that causes all kinds of complications from this disease. So let's talk about how these changes, like the change for sickle cell anemia, come about. We talked about proofreading just very generally in chapter 13. As part of the process of DNA replication, one of the DNA polymerase enzymes goes back and actually proofreads the DNA and looks for mistakes. And occasionally mistakes occur. But the job of this particular DNA polymerase is to make sure that only a limited number occur. It wants to correct as many as possible. Even though the proofreading activity of DNA polymerase is actually pretty good, we still have an error rate of about 1 in 10 billion nucleotides. So every time DNA replication happens, actually some mutations occur in the DNA. If a mutation occurs, hopefully the DNA polymerase corrects it. But if not, there's a mutation in the cell. And this is a really important source of new genetic information. So we often think of mutations as bad, and generally they are, because if you think about it, an organism is already adapted for its environment. And so if it acquires a mutation, then it's probably not going to be adapted. But think back to the genetic code. There is some room for mutation in the redundancy of the code, in the wobble of the code, and so not all errors are actually detrimental. So errors occur in DNA replication, and that is actually an important source of new genetic information. But errors can actually occur after replication as well. So these errors are often the ones that we see that produce cancers and things like that. A mutation from a genetic standpoint is important only if it's passed on to the next generation. So if you have a change in a gamete when DNA replication is working in the process of meiosis, if you have an error there, that error is going to be passed on to the next generation. And those changes in the DNA are what is going to drive evolution in a population. Because now you have a new genetic variant that has come about. So mutations are important because they can allow for new variation that evolution can work on, but they are also important because they're the only source of new genetic information. We talked about genetic variation and meiosis and crossing over and independent assortment and fertilization due to sexual reproduction. Those mechanisms increase genetic variation. They shuffle variation, they move variation around, they produce new combinations, but they don't produce new variation. Mutations are important because they produce new variation. And if those variants are important in the environment, those variants will increase in frequency. If they're not, if they're detrimental, the organism is probably not going to survive to pass on its information, and so that mutation is not going to stay in the population. But if it is beneficial, it's going to stay in the population, and that population is now going to be a little different from any other population around it. Oh, I don't see a slide. I had a slide in here that talked about what happens or when errors occur in your somatic cells. So we think about mutations as those that are passed on, but you can also acquire mutations due to environmental exposure. So things like UV light, cigarette smoke, asbestos, different chemicals in the environment can also cause mutations in the cells. And those mutations quite often lead to cancers. When you have mutations in the cell cycle controls, you often the individual can develop cancer. It's not to say that those are not important, but from an inheritance perspective, those ones are not passed on to the next generation. So if you're talking about genetics and inheritance, we're interested in the mutations in the gametes. Of course, those mutations that occur in the somatic cells are also very, very important to the health of the individual. So let's talk about some of these types of mutations. There are two main types. There are single nucleotide substitutions. So these are where one base pair is changed for another. And the other one is insertions or deletions. So let's start with single nucleotide substitutions. The first type that we're going to talk about is silent mutations. These are also called neutral mutations. I'm fine with you calling them either one. I tend to call them silent, but if I say neutral, they mean the same thing. A silent mutation is a mutation that does not change the amino acid, and this is due to redundancy. So if we look at the sequence here, we have methionine, lysine, phenylalanine and glycine. And if we looked at our parent sequence, we have a G in the template strand, but if we change it, if a mutation occurs and we switch to an A, we actually just have some wobble going on. So we still code for a glycine. So that mutation has happened, but it hasn't at all changed the sequence of the amino acids, and so that mutation doesn't show up as a change in phenotype. We can also have missense mutations. So a missense mutation is a mutation that causes a change in amino acid. So here we've got a mutation at the first base pair in the triplet code for glycine. So in our DNA template we have CCG. We've substituted now a T for that first C. That causes a shift in the amino acid from glycine to serine. Now, depending on the amino acid, this may or may not cause a big problem. If you substitute something similar, say an amino acid that has some acidic properties for another one that has some acidic properties, you might not really change the function of that protein. But if you change it to something that causes a problem in the shape of that protein, you can very quickly get a completely dysfunctional protein. Or a protein that might fold and things, but it's not going to do its job in the cell the way that the original template strand wanted it to. Finally, we can have a nonsense mutation, and this is where a mutation is introducing a stop codon. Now, this can happen at any amino acid along the way, and when that happens, it affects everything after it. No more amino acids are added, and so your protein is essentially truncated, missing a part of its functional structure. When that happens, you're absolutely not going to have a functional protein. So nonsense mutations are very detrimental to a phenotype because you get a dysfunctional protein. With missense, you may or may not get a protein. In the sickle cell example, that was a missense mutation where you change the amino acid, so you still get a protein. It's just not functioning the way that you expected it to. But with nonsense, you lose part of your protein and it cannot function the correct way. So we can also have insertions or deletions. So this is where a single or multiple base pairs are inserted into a DNA sequence. Now this is a problem because it causes a frame shift in our amino acid sequence. So remember we have our reading frame, and I did those examples at the beginning, and you might want to go back and just review that slide where I show the fat red cat sat and the impact that reading frame has on the sequence. But if we look at these three examples here, these are the three different ways that a frame shift mutation can occur. So top right, we insert an A. In this case, we have produced a nonsense protein. We introduce a stop codon, so nothing happens. Because now we have an extra amino acid, it shifts the reading frame all the way down, and we introduce a stop codon. Over here on the left, we introduce a random base pair, and it changes all of the amino acids after that. So we start out with methionine lysine. We should have phenylalanine, but instead we have leucine, and then you can see the sequence changes after that. Again, that's because we've shifted our reading frame by one letter. We've caused all of our words to change following the amino acid insert. And then the same thing can happen with a deletion. If we remove a segment, it might be one amino acid, it might be two, it might be three, but we are losing genetic information. And so that's, again, causing a change in the amino acid sequence, which is going to produce a very different protein and could potentially produce no protein at all. Here, when we actually, never mind. I was going to say we introduce a stop codon. We actually don't do that here. But your insertions or your deletions along with your single nucleotides can all produce missense or nonsense depending on the situation. And then of course we can have silent mutations, but that only occurs when you have a single base pair change. So let's look again at sickle cell. So we have the wild type DNA sequence here for the normal subunit of our hemoglobin. So we've got our DNA, our mRNA, and then our amino acid here. Here we have had a missense mutation. We have substituted an A for a T, which gives us a different triplet codon and therefore a different amino acid. All right, so during our genetics unit, I asked you, or many times discussed, sources of genetic variation. We had independent assortment of chromosomes, so different combinations of mom and dad's chromosome ending up in the gametes of the individual in question. We also have crossing over, which is where individual chromosomes, homologous chromosomes, actually exchange information, so we get new chromosomes. And then we get a lot of genetic variation from random fertilization. So a random egg is fertilized by a random sperm, and now you have new combinations of genetic material that you never had before. So let's add a couple of more things to our genetic variation. When we were talking in chapter 10 about non-disjunction and chromosome structures, you can also get genetic variation due to chromosome abnormalities and changes in chromosome number. One of the things that's interesting about the human and the chimp genomes is that the chimps actually have one more chromosome than we do, but the information is very, very similar. And what has happened is that one of the chromosome pieces has moved within our genome, and so we have different chromosome numbers, different chromosome makeups than chimpanzees, although a lot of the information is very, very similar. And then, of course, we have our chromosome abnormalities, our non-disjunction disorders with Down syndrome and Turner syndrome and things like that. Finally, our last source of genetic information is mutation. So remember, mutation is the only source of new genetic information. All of these other ones just sort of reshuffle and redistribute our genetic information in a cell.